The mission of Trailhead Biosystems is to deliver cells of any type and hereby enable specialized cells for the uses of disease modeling, therapy development, and drug discovery. A snapshot of the company is that we got a couple of years on our back right now. We're founded in 2015 through the state of Ohio, supporting us to build a platform that can perform very, very large dimensioned experiments so that we can explore the differentiation space of human cells as they go forward in differentiation. We have since then developed this technology into multiple areas of stem cells, including MSCs, HSCs, pluripotent stem cells, and a couple of others that I don't have to go into. The data that we're generating are all built of in-house generation of data that we can then explore and mine for the purpose of developing specialized recipes that give an improved level of differentiation of the human cells. I think we might be the world's first developmental biology geared company. And the main differentiating aspect of Trailhead Biosystems is actually that we got a remarkable ability to, at low cost, develop processes for the media that goes in when you need to control differentiation or phenotype in general in a bioreactor. We have already now filed several lines of IP on the various cells that we're producing. I'm going to talk about a couple of these. I can't show them all. You see the particular areas of biology that we're entering into already. One thing that I want to mention is that we actually see an emerging data economy from doing these very large experiments, and that is that we are equipped with the same genes in all our cells, but the phenotype is different. So when we learn something about the behavior of a gene in one particular phenotype, we can usually use that in another phenotype as well. And that gives us the ability to even further accelerate the discovery of the exact process inputs that control the cell fate. We're also very broadly partnered. We're very happy to be partnered with in BioFab USA through ARMI, the Advanced Regenerative Manufacturing Institute, and many of our programs are related to activities that they partner in with. We have recently been capitalized. We're right now around 20 full-time employees in Ohio. The way it actually works, if you look at it, is that when you develop a protocol at a bench scale, you're usually doing it a hypothesis-driven way. A person comes in with an idea, tests this hypothesis, gets data, and incrementally moves this forward, but never explores that space of differentiation in the way that the cell senses it, which is always multidimensional. What we're doing instead of this typical bench-driven, hypothesis-driven research is that we're empirically testing a very large space at the same time, usually 12 dimensions of inputs, so that we can statistically qualify every single time that a particular factor is interacting with another to elicit a desired outcome, say, for instance, that the cell differentiates into a lineage that we're liking. So just to make it sure that you know that we're not really mining other people's data. Yes, we like scRNA-seq data sets. We can align up to look for the states of expression in such data sets, but we're not actually using that to understand how to control a cell fate. We have to generate the experimental data in-house to understand how the cell actually behaved. So we are what we call empiricists. In other words, we are scientists that uses, in this case, robotics to execute the experiments and computers to design the experiments, because no human mind is equipped to actually do that. So when you look at what features that then emerge for the protocols that are now coming out from doing this, is that they stand remarkably different to what you usually see when you look at a paper published on how to generate a specialized cell. So as common of these features are listed here. We have decided to simply always go back to the beginning. We built these protocols up from scratch. Why do we do that? That's because we are not actually believing that the first stage of differentiation by any publication necessarily is the best. So therefore, we are exploring that space in the published literature to see if we can find and validate what we already know, or if we can enhance it, which we have actually done in multiple cases. So we build them from scratch and we move forward and we end up getting a multi-stage protocol coming out of this. If it's different, we file for IP. If it's the same, then of course we have to do a freedom to operate analysis of that. The protocol is always data-driven. It's not hypothesis-driven, as I was saying. It means that we're less cognitively biased towards wanting to work on something that was already based off what the company did. We can identify this thing called critical process parameters, which is a thing that any process engineer actually loves to know, because if you know the process, the critical processes in the CPPs in a process, you can focus on measuring those and keep them within a certain range so that the process remain robust and stable. And that's what I think we need to do in the bioreactors, but you need to know these parameters within your media. And if you haven't actually calculated them, you are unable to know whether you're dealing with an agonist of a pathway that is actually a CPP. Quality by design is a principle that stands behind the application of this type of approach. So it's part of the guidelines to industry, especially in the pharma industry, but I think we will see it increasingly going forward that our industry in the cell therapy area would be more and more leaning on common principles on quality by design. We need to validate these cells, so we usually go in with molecular signatures, RNA-seq, as well as molecular signatures because they are common, so I can show you some pictures of that for the various results. We like to stop development at the time when we have attained what we call homogeneic conversion. In other words, that the cells that we started with homogenetically, or in a homogeneous way, converted into a desired state. And we have then found that there is a certain amount of universality here because our media then performs better when you start with different IPS lines from different donors because the media simply has a higher capability for controlling fate. And one additional feature that I like to bring out that we're actually finding that we're quite good at replacing expensive molecules with cheaper ones. We are now very, very cost-focused in the company because we have found out we can and we think it's necessary. Many media that you use include a lot of very expensive proteinaceous components. They are, of course, not only expensive, but they're also a little painful to work with because their diffusibility in a complex tissue, such as an aggregate, is diminished because they're usually referred to what we in developmental biology call morphogens. They are diffusion restricted. So it can be hard to get them to touch all the cells if you, for instance, run a bioreactor aggregate. A small molecule is more stable, better able to penetrate and exert its effects. It's also a lot cheaper. So if we succeed in getting a small molecule replacement, we're very happy. And we can then transfer that into our bioreactors to see if we can convert this into a less expensive process. Inside the company, we've decided to split this up like a developmental biologist would into the 3 germ layers for which our cells descend: endoderm, ectoderm, and mesoderm, which of course corresponds to the starting point for all the lineages in the human body. And we now have individual groups operating in the company that takes care of the protocol development within these areas. So we have a leader for our ectoderm program that performs the protocol work for the neuronal cells that we make. We have another one that deals with the work on the endoderm, and then another one for those of the blood lineages that we're doing. We address multiple markets. We may be the smallest company that addresses the largest number of markets with what we do. We may be too bold to do it, but we do it because we know we can, and we have a rather low cost and time involvement in building a protocol up. So as you can see here, we have in the iPSC space, we have various neuronal cells that are built up to be improved versions that might have a better clinical outcome when tested, that address various diseases, as you can see on this slide. And then we have various programs that address needs mainly in the hematology area. And then amongst them sits the emerging field of cell therapy for diabetes, in which I have been an investigator for many years. Here is a little overview of the current pipeline of the most prominent programs we have, according to red, mesoderm, green, ectoderm, and blue, endoderm, and where we are at, and the indications, what the origin of the cell is that we're starting with, and how far we are. There wouldn't be a program on this slide unless we have the confidence that we can make that cell and that we've validated it, but we may have filed or not filed IP on the process, and now we're moving into reactor validation of these protocols. So this is a standard way. We need to validate the cell, show that we can scale it, and then we want to deliver it further. I have 3 programs that I can show some data for you for. One of the exciting ones is actually through interacting with ARMY, of generating cells of the CNS, and in this situation we're doing this actually with ARMY, Biogen, and Arizona State University. One of the cells we're building that I'm going to show here is the oligodendrocyte, and I'm showing that because we have built a fantastic protocol up that is very short and very robust. And it is because the literature on making oligodendrocytes is that this is a mess. It takes a very long time, and you end up having a very heterogenic population with a mixture of oligodendrocytes and neurons. But we start out here to immediately get into the OPC progenitor state of pluripotency. The protocol is very different from what you see in the literature. We can then go on in approximately 2.5 weeks to get rather homogeneous induction of the very late stage myelinating markers, such as you can see here, the CNP gene for myelination, and then your PLP, the proteolipid protein 1, which are the back-end terminal markers for the oligodendrocytes themselves. And they still express the markers that we... And these are, as Blue Rock would say, authentic cells. They haven't been genetically edited. They are simply induced to this fate through a controlled differentiation process. So we're very happy having now oligodendrocytes that we want to scale, and they can be used for drug discovery and, of course, several other things. Here is one example slide from a blood project where we, as you know, it is possible to actually make erythrocytes from HSC through published literature, but still we have to bring down the cost tremendously to make it feasible to make such a scale. In order to do that, we need to attain a much more robust and direct, rapid way to generate the enucleating erythrocyte. So we're here comparing a protocol that has 3 stages. The first 2 stages have been developed, and I can show you the results of that, where we very quickly actually move up to a state that expresses GATA1 and then the transferrin receptor, and then moves on to express glycophorin A and the hemoglobins. And we can compare with this published protocol that is more expensive on the efficiency of this process. How quickly can we walk the cell up to the state we want, and then further into next stage, so we simply follow the standard differentiation schema going through the usual megakaryocyte erythrocyte progenitor cell onto a committed erythrocyte, and then to get an enucleation process going. If you don't have the media that can do that, your bioreactors won't perform, and your eventual packed red blood cells will not really be up to par. So here is one last one, and that is on the beta cells. We did the same thing. We started from scratch. Everybody knows, most people know, the diabetic field started with de Moor's paper in 2005 for making beta cells out of embryonic stem cells, and we're now plus 15 years in. And we still don't have a protocol that's really that good. We wanted to see if we could build a protocol up. We found a way to actually make what's actually called the dorsal pancreas. It is so different from the published literature. There's no activin use in this. It is not a definitive endoderm induction with the standard Wnt and then a DE inducing step with activin. This is entirely small molecule and effectively generates a pool of cells that we qualify as dorsal endoderm that then takes on the pancreatic fate. And then, just like the ventral pancreas goes on to develop very quickly if you roll them into differentiation pancreatic beta cells. So right now, we, of course, as everybody else, we're working on finalizing this protocol to bring up the metabolic entrainment of these cells, increase the number of beta cells to in vitro develop functional islet equivalents. And that's, of course, the destination of this program. So as we just heard from Aspect, the idea is to replace islets with a stem cell-derived source, and there are many companies that are now engaged in this space. We're as interested. Program remains unpartnered, but inside, and we need to find a way to get these to the patients. Comparing the protocols, I just made a little matrix so that you can compare for the 3 that I'm deciding to show to you today. For the oligodendrocyte, we are dealing with a rather short tissue culture protocol. So we save time, we save cost, and again, we get a homogeneous differentiation process working because that's what we sought to build. And it's a much, much, much cheaper protocol compared to what you would do if you pick a literature center piece publication as the one we're listing here, which is the one we're benchmarking against. Same thing on the red blood cells, but at this point, that protocol is not as cost-reduced as the one in literature, but it still performs better. And then we have the insulin-producing cells that, by virtue of this being small molecule only, is actually dramatically reducing in cost compared to the standard ventral pancreas method based on activin. So of course I'm now super interested in getting iPSC at scale at the company, which we're doing, so that we can take those and bulk differentiate them forward at low cost. And it's my hope and expectation that within the next year, perhaps at the next meeting, we can talk about our offerings of in vitro derived beta cells for you. Would very much like to entertain discussions on what we should do with these going forward. The team is rather large already. We have a fantastic team of individuals that are doing this work. And we're furthermore very pleased with the fact of the outside advice and guidance that we have available. The company is, as I said, is located in Cleveland. We expect to have some presentation on either side of the coast in the US to get closer to people that want to partner with us in the area of any of these cells. As I said, we can make any cell, and we are very interested in learning about which cells that you may be interested in, so please reach out. Thank you very much.